Cool. Hang on,
John,
let me just
do you want to
share. Yeah.
share your screen? Thank you.
Can everyone see that? Yep, cool. Hi, I'm John. I'm a second year graduate entry medical student. And before medical school, I did a master's in immunology and worked as a research assistant in immunology at the Jenner Institute, where I worked on malaria and outbreak pathogen vaccine clinical trials. So it's apt that I talk about vaccines today. I'm going to outline a bit. I'll probably overrun because of the kind of person I am, but sorry. So I'm going to run through a quick history of vaccines, the principles of vaccination, different types that are available, and then I'm going to move on specifically to clinical trials and talk a little bit about COVID trials and why they haven't taken as long. And then I'm going to focus on two specific vaccines that are sort of the more hot topic right now for the UK. I like putting this slide on whenever I talk about vaccines because I think it's understated just how important vaccines are in the general public. People take it for granted, but the World Health Organization, estimates that almost a quarter of deaths worldwide are due to infectious diseases. And shockingly, for the under fives, over half, just under 65% of under fives die due to infectious diseases. Vaccines are incredibly cost effective and are arguably the most powerful public health tool ever created. I'm sure some of the public health doctors would probably agree with that. So can anyone tell me who this is in the chat? Really easy if you've got a keen eye. Oh wait, I actually can't see.
You can see his name next to his chin.
I just realized I can't see the chat either, sorry. Unless I tab out. Yeah, so this is Edward Jenner, known as the modern father of vaccines. And can anyone tell me what this is? I don't know, I've just asked again. I'll just move on and just say what it is. So this is smallpox manifestation, and Edward Jenner was a physician in Gloucestershire, and in 1796 he noticed that milkmaids were not getting infected with smallpox, which was endemic at the time. So what he did was cut into a smallpox lesion from a milkmaid called Sarah Nelms, and inoculated a child called James Phipps with it. He became unwell, but upon reinfection with a smallpox pustule material, he didn't develop smallpox because of Edward Jenner's theory. You would absolutely never get away with this in ethics today. But it gave us the first documented case of vaccination all the way back in 1798. And just to hammer home just how important vaccines are, if you look, this is who's, oh wait, hang on. Laser pointer. This is Public Health England's infographic on vaccines. Diphtheria used to affect 50,000 people in the 40s, and in 2014 there was one after the introduction of vaccines, so a 50,000 decrease. The same thing with whooping cough, measles, Hib, and Mense. All of these go down, and some of these are completely devastating diseases, especially for childhood. And this is just a nice little roadmap of where we started all the way back in the 1700s with Edward Jenner's vaccine on smallpox, all the way through to polio, all the childhood vaccinations, cholera, MMR, all the way to the modern days. So we've come a long way for our vaccines, but the impact on public health is absolutely monumental. So the principles of vaccination. So vaccination is the controlled exposure of antigen to generate what we call immunological memory. And this is part of something we call the adaptive immune response. So the adaptive immune response generates a specific immune response to a particular organism. So it's highly specific. And the key feature is that once it encounters a microorganism, it will remember that, so that a second time you encounter it, the response is faster, more targeted, and much stronger. So there's two parts of this. There's something we call humoral immunity, which essentially means antibodies. And these are produced by a cell called B cells. The other part is cell-mediated immunity, which is due to cells called cytotoxic T cells. And these release various molecules and enzymes that directly destroy virally infected cells. I'm going to be talking about it in the context of viruses rather than anything else because it all gets very complicated. And a little bit more on antibodies. They can either bind directly to the virus and stop the cell from being infected in the first place, or bind to infected cells and cause either clearance or inhibition of all these viral techniques that replicate and pass on the virus to other cells. So other types, so there's a lot of types of vaccines. The first one is live attenuated. So attenuated essentially means deactivated. So these can replicate or cause severe, well, they can't cause disease in people. And then there's whole inactivated and split inactivated. So these are usually heat killed, and they quote unquote kill the virus, even though viruses are technically not alive. And most vaccines on the market that are licensed are these three here. The next is synthetic peptides. These are just little bits of protein that you inject in people. I don't think they're very good, but they still exist. There's something we call virus-like particles. So these are synthetically made molecules that resemble viruses but are non-infectious, and they don't contain viral genetic information, so no DNA or RNA. And essentially a way to induce protein expression on these particles that triggers an immune response. And then there's newer, and recombinant subunits are essentially the same as peptides, but are whole structures rather than cut up bits of protein. There's also two newer technologies, the DNA and RNA vaccines and the recombinant viral vectors, which I'm going to talk about this one a bit, RNA and recombinant viral vectors a bit later. But just to note that the DNA one uses a special kind of DNA called cDNA generally to induce expression. And we use that in science all the time. And it's quite safe before people get up on that. So clinical trials, what makes a good vaccine? So a good vaccine should be safe. It shouldn't cause any adverse reactions. Most of people get, a bit sore arm. It might get a bit red and hot, but that usually subsides. And there's vigorous testing about safety in vaccines. So a good vaccine will be protective, and the absolute best vaccines will induce a T cell and B cell memory, so humoral and cell mediated memory, and ideally long-lived protection. So you wouldn't have to get, a yearly vaccine like the flu. And practical considerations, we want to keep costs down, especially considering a lot of infectious disease worldwide affects low-income countries that don't have a lot of resources. So we really want to make sure it can get to everyone to have the most global impact. Delivery, some of them aren't very stable, so how can we get them from the manufacturing plants to the general public? We have to consider storage. This has been a big thing at the moment, with how we're going to store -80 mRNA vaccines versus vaccines that you can store in a 4-degree fridge, and side effects. You want to keep these down, obviously, because it's just not very pleasant having side effects from medication. And we also have to keep in mind that every infection is different, and each one needs specific considerations depending on who you're targeting. For example, malaria is especially important in young children and in resource poor settings, so you need to make sure it's available to them at an easy way. Whereas you compare that to something like the prostate cancer vaccine is easier to give out because it's more prevalent in more affluent countries where there's a lot of resources. So I'm going to talk a little bit about, well, I say a little bit, a lot about the process of clinical trials, and then I'm going to talk about COVID clinical trials in particular. So this initial part, design and exploratory preclinical studies, is done in animals such as mice, rats, ferrets, non-human primates, all of these 2 stages. And it involves the sequencing of your target and then exploration to find vaccine methods and to generate your vaccine. You then move on to preclinical studies and toxicology studies. So these are to test the efficacy, the immune response, and safety in animals before you move on to anywhere else. These take around 2 to 4 years. These can take this is such a variable stage as well. Most of them fail at these 2 stages. Most of the vaccine candidates. And toxicology studies are extremely important for the rest of the phases. So you then submit something called a investigational new drug application to regulatory bodies. And once they say, yeah, your data looks good, you can move on to human trials. So phase 1 trials are first in human trials. And these are usually small numbers, like 30 to 100-ish. And the primary endpoint in phase 1 trials are safety. And you can also add in immune responses and dose ranging, but safety is the key one that you need to move on to phase 2 trials. And phase 2 trials specifically look at efficacy and immunogenicity with slightly larger numbers. And you look at the effectiveness in treating or preventing the disease. And you also look at dosing to determine a therapeutic dose. And then once you finish these, you can say, yep, this vaccine has efficacy, and you move on to phase 3. So these are much, much larger trials. You can have thousands of participants, and they, again, look at efficacy, safety, and immunogenicity in larger populations, and to determine its therapeutic effects compared to other therapeutics for that disease. And you assume at this point that your vaccine has a real efficacy. Following this, you can submit a biologics license application to regulatory bodies. And the main one in the UK, which has been in the news a lot, is the MHRA, the Medicines and Healthcare Products Regulatory Agency. And that's the main one. There's other ones like the FDA in America. But once you're approved, you can then start large-scale production and distribution. And then once this has occurred, you also undertake phase 4 trials, which is post-market surveying in the public to monitor long-term effects. And it's very, very important to highlight that at every stage, safety is heavily considered and documented. Any safety concerns are followed up and trials are stopped for this reason. So an example of this is during the Oxford trial, a patient developed transverse myelitis, and the entire trial was put on hold while it was investigated and determined that it wasn't due to the vaccine. And this is standard practice that a lot of people got a bit upset about, but it's what we do to ensure it's safe. So why was the COVID trials shorter? So this stage was very, very shortened because we had existing data on SARS and also MERS, Middle Eastern Respiratory Syndrome, which are both coronaviruses. So much of the discovery stage was skipped. And we also had pre-existing vaccine technologies, which we knew were safe and efficacious. And the sequencing was done within 48 hours by someone called Professor Zhang Yongsheng in China, who immediately made it published online. And that was just an absolutely monumental effort and sped up the entire process so quickly. So good job to them. So yeah, much of this was skipped. And then the regulatory bodies were extremely quick in approving all this data, given that it's not given the usual time frame. And several of the phases, phase 1 and phase 2, happened in parallel. And to speed things up, no corners were cut though. This was most vaccine clinical trials places put every other trial on hold while they were doing this. And when the interim data from these 2 phases were available, they then submitted those again, and phase 3 were allowed to undergo. And another thing that really, really sped it up is that production of the vaccine was started before the phases and continued throughout. And this was at a risk to pharmaceutical companies, because if it didn't work, they would have lost on a lot of money. And also emergency use authorization by regulatory bodies speeds everything up as well, which is why rather than 15 years or longer, it has only taken 10 months to 1.5 years. There are 2 vaccines that are currently licensed, one in the Chinese military and one by the Gamaleya Research Institute in Russia. But the Russian one was licensed without a phase 3 trial and is dubious at best, in my opinion. So I'm going to talk about the Oxford vaccine. So I've worked with ChAdOx before, ChAdOx 1 and ChAdOx 2. I think they're very good. It's very good, the technology. And they were produced by the University of Oxford and AstraZeneca. So this is a coronavirus. And the thing that we're very interested in is the spike protein, which is expressed on its surface. So we use a chimpanzee adenovirus as a viral vector. And adenovirus is what causes common cold. And we essentially take the genetic material for the spike glycoprotein and splice it in to the DNA of the adenovirus, and then give that to patients so that the cells can express the spike protein and elicit an immune response and induce immunological memory. So why do we use chimpanzee adenoviruses as a vector? So they can be disabled genetically. So the virus we use isn't able to replicate within the human body and cause disease. And it's an excellent way of introducing foreign antigen into patients. And the vaccine doesn't require anything else to like boost its immunity like protein and subunit vaccines usually do. So a bunch of them have things called adjuvants in, like aluminium. You don't really need that. And it also induces a very strong T and B cell response because humans have minimal exposure to chimpanzee adenovirus as well natively. So it's just very good all around, in my opinion. Yeah, and that's it. Oh, I should also mention that the reason it enters cells is because the spike protein, why it elicits a response, because the spike protein interacts with ACE2, and that can also help trigger the entire process. So the second vaccine is produced by Pfizer and BioNTech, and this is an mRNA vaccine. So a bit of background. So DNA is essentially the blueprint for proteins, sort of like building blocks of the human body, but it's too precious to leave the nucleus, the sort of brain of the cell. So you transcribe it into something called mRNA or RNA that can leave the nucleus and do whatever it wants. So mRNA usually goes to these structures called ribosome, these little dots here just outside the nucleus, which are thought of as factories that produce proteins. So in this vaccine, you take your spike gene for mRNA, sorry, and then it is encased in a lipid nanoparticle, which is just fat, and they can dissolve into cells, and the mRNA goes to the ribosome and tells the cell, hey, start producing the spike protein. And then this is expressed on host cell surface and elicits an immune response. And there's a lot of like myths about RNA. So I've seen a lot in the media saying, oh, will this not mess with my DNA? And no, it simply doesn't go into the DNA at all. It completely bypasses it. So that's not a safety concern. And I found this nice little comic that I thought was quite cute. So I'd wanted to put it up. So that's my 20 minutes overrun. Thank you so much for listening. And I'm happy to take any questions.
John,